Good morning, everyone. So today, I would like to take the next 10 to 12 minutes talking about one of the groups that we tangentially touched yesterday a little bit, these virophages, and I'll let you know the different nitty-gritty details about how we are using our different techniques to discover these viral entities that they are very poorly discovered and described so far using global metagenomics. So basically, the idea of the talk will be giving a little background, a brush up of your knowledge about virophages, talk a little bit about the discovery, the discovery pipeline that we have used for these entities. We'll talk about the diversity, the huge diversity that we have found using major capsid proteins as a bait for discovery of these viral entities. Also, we'll talk about the wide habitat range in which we found these viruses. We'll talk a little bit about the genetic repertoire that we find and how these genes are shared across different other viruses. Finally, I'll let you know how we are better describing or completing more the collection of virophages that they are out there right now. I'll let you know about the list of complete and near complete newly discovered virophages that we'll have. And hopefully, unfortunately, I won't have time to talk about our computational approaches to predict some hosts right now. So basically, what are these virophages? So they were discovered around 10 years ago or so, and they are double-stranded DNA viruses. They are mid-range in size from 15,000 to 30,000 base pairs. There are so far only 3 non-redundant isolate genomes. These 3 guys over here, Sputnik, Mavirus, and Samilon. And depending on the different studies that you can find in the literature, so there is a number between 30 to 50 metagenomic virophages. So that depends if you are considering full length or near completeness of the genome. These are mostly found in aquatic environments because they are associated with their host, mainly in freshwater sources, but they were also found in marine habitats and also in soils, and they are predicted to be found also in some other animal guts. They are able to replicate in their final host, which is a protozoan or an algae, along with giant viruses. So they basically use these viral factories inside the eukaryotic cells in order to replicate with their giant virus that they infect. And they are phylogenetically related with polyntone viruses. And if you were here yesterday with Dr. Koenig's presentation, so here you can locate them in this particular region of the evolution of viruses. So the important point here is that for us in our discovery pipeline, al contrary, as Simon's presented today with innoviruses, so these particular entities, they have a very conserved capsid that will help us to the discovery of those guys. So basically what we have done is taking an advantage of the large database that we have in IMG. It was to use at that time, 17 major capsid protein genes that they were in the databases, and we search against IMGVR using just a blast P of these proteins, and we only found a very number of hits to them, and we decided not to use only these hits, but use the result of the hits that we found to combine with the number of major capsid proteins in order to develop some models, actually 4 different models that we subsequently used to mine all the 10,000 samples that we have in IMG. So once we did that approach, so we collected a large number of novel major capsid proteins that we, again, used to combine with everything that we knew at the beginning And we regenerated our models, and instead of having 4 models, so we ended up having 15 different models. So then with the 15 models, so we went back and mined our database in IMG, and also we mined a second database that we obtained from the SRA that they were related with human gut. So it's around 4,000 samples that we assembled, and we mined our models against them. And finally, with these 15 models and the 2 databases, so we ended up having a collection of around 45,000 non-redundant contig, and around 500 of them, so they were larger than 10K bases. So I just want to take a second talking about the importance of this connection with, double iteration of the models, because now we were able to assign some models to some particular habitats that we never found virophages before. So, so here you have the models that contain any of the members from known virophages. And then, for example, you can see that for these 2 particular models, the family 5 and family 7, so now they, we found a bunch of different hits across many different environments, and particularly these that we were very much interested, like the human body. So they were not able to be found using any of the models that they contain any of the references. So this is an important point to point out. So what did we find? So when we used this approach, I told you that we had these 45,000 unique contigs. So these unique contigs contain around 30,000 non-redundant capsids. So this is the tree of the capsid. This is the diversity of the virophage. So basically, you find a large number of novel branches, and you can see much here. But if we collapse the tree, what you can observe is that we are vastly expanding whatever it was known before, and we are finding novel virophages in many other different clades. So basically, this tree is the same one that we had before. In red, we have the location of the previously discovered using metagenomics virophages. In gray is everything that we have added to the tree, and in blue you have the location of the 2 major capsid proteins of the isolates. So it's a huge expansion in the capsid diversity. And also, if we look at the different habitats in which we found these virophage particles, so you see that obviously the dominant environments are still aquatic, because they rely on their hosts, and the majority of the proposed hosts are algae and they are protozoan, that they could be related with these aquatic environments. But we also found a large number of hits against some novel habitats, like host-related habitats like arthropods, humans, and cow rumen, and so on and so forth. So I just want to take a couple of minutes to talk about our discovery in the human gut. So basically here, we were very confused, because when we used for the first time our models, and we used these 15 models against IMG, so we were unable to find any hit to any of the human gut samples that we have in IMG. And we were a little surprised, because there is no reason that apparently that we cannot find these entities on the human gut. But then when we use a separate database, that this is coming from the SRA, that is more rich in terms of variety and metadata and related with the samples. So we found a clear pattern that apparently these virophages are actually correlated with rural environments. So that means that if we have samples from urban styles, so we basically don't find any hits to this major capsid protein. And then, on the contrary, if we use any of the rural habitats for this database, so we found that the majority of the Fiji, El Salvador, and Tanzania, for example, countries, so they contain hits to this particular viral groups. And also, moreover, so we found that there is a clear trend to some specific of models that we found in this particular area. So when we detected something in the urban areas related with these virophage particles, so they were based on one model, basically, and totally different models if we find then in rural habitats. And out of these guys, so we complete and we close and we have some insights about these particular genomes. So talking about what is the repertoire of these virophages, so if we take only the fraction that I told you of these 500, around 500 contexts that they were larger than 10 kb, and also some reference, I mean the original references and some other references, entities that they are related with these virophages. So we've and we created a data set of around 10,000 proteins that we collapsed into around 700 super protein clusters. And what we observe is that there is only a group of 4 core genes that they are represented across pretty much all the viral, the virophage entities. So those are related with the structural proteins, the major capsid protein, minor capsid protein, a packing, ATPase, and a protease that seems to be related with the structural genes. Then we have a second group of genes that they are represented in between 25 to 40% of all the genomes, and then a large tail that they are not very commonly represented across all virophages. So with this in mind, we took these 4 core genes in order to predict which ones out of the collection that we have in here were either closed meaning that they are circular, or they contain at the very end these inverted terminal repeats that they were shown before, or near complete if we don't have the circularity for some of the virophages, but they contain, I mean, they have a size over 10,000 base pairs, and they contain 4 of the core genes. So just to finish, so this is our idea of how can we now represent this group of virophages. So basically, so we are using phylogenetic trees, only using these 4 core gene-based tree, taking all the genomes that we have from our collection, plus some of the references that we have. So we can see that there are 7 clear new groups based on these 4 core phylogenetic tree. Very interestingly, so we can see that they are actually not only clustering based on the 4 core genes, but also there is a clear synteny of these 4 core genes. So we can see that we are expanding dramatically some of these branches, and also we are presenting the synteny of the 4 core genes. Also, there is a very interesting relationship with some of these groups with certain habitats. For example, we have here the rumen-like and the human-like groups. So those are a single branch that they were found exclusively in this group with this particular synteny. We have some of the groups that they are present mainly in fresh water, some of them that they're only marine and non-marine but saline environments, and so on and so forth. And finally, we also can have an idea of how these genomes vary in terms of their size, so per clade. So we can see that some of the groups are clearly longer sizes than some others. And also something that is interesting, but we don't have an explanation yet, is that apparently there is no correlation between the genome structure of some of these virophages based on circularity or the presence of inverted terminal repeats. With that, I just want to finish with our conclusion. So basically, in some cases, like these virophages, it's very interesting to develop these models. In our case, so we did 2 iterations, major capsid protein model development, and that allow us to correlate some of these new models with some habitats. Some of these habitats were very unique. So for the first time, we discovered full-length, complete virophage genomes associated to humans, to arthropods, and to some plants. We are confirming that there is only a 4-core set of genes for this particular group. Finally, we have closed 92 complete virophages and 241 near complete based on our approach. And we are proposing that we can classify these entities right now using this synteny and this concatenation of the 4 core genes tree, proposing 13 new groups, 13 groups, 7 of them being new. So with that, I just want to thank everybody involved in this project, especially Jingle Su, plus all the JGI viral community. So thank you very much.